National Evolutionary Synthesis Center and Understanding Evolution, this is the Evolution in the News story for November 2008. I'm Kristen Jenkins. When we hunt and butcher game, or live in close quarters with domestic animals, we are exposed to the pathogens those animals carry, giving pathogens the opportunity to try out a new host. In some cases, the pathogen is able to successfully infect human hosts. In other cases, the pathogen fails. Understanding how and when pathogens in the environment evolve to become human pathogens can help us better understand and control the diseases we have now and prevent future transfers. Dr. Katya Cole, an assistant professor of biology at Duke University, studies the evolution of infectious diseases. In this interview, she talks about some unexpected information we can learn from reconstructing viral histories.
So viruses can be very useful in understanding or piecing together historical events because viruses very, very rapidly mutate. And so if we have a sample of a viral population, we can understand certain things about the population that it infects. So I want to provide an analogy of how this actually works first. So you can imagine some kind of relay race in New York City, in Manhattan, and the race starts with it's a very unorthodox race, and it starts off with just one person at the starting line, and that person has this baton, and that person can replicate its baton and hand it off to all of its friends that go running in different directions and do the same. So over time, the people who are in the race, there are more and more people in the race, and they're also all over the city after a while, or spreading from where the race started. So you can imagine this as an analogy to viruses, where at a specific point in time we can sample the viral population, which are kind of like those runners, and from knowing what the genetics are of that virus, we can use that information to get an idea of when and where really this race started. So when did that first? Where was that first runner? Where did that infection actually start? Michael Warabee's work on and his colleagues' work on HIV was really groundbreaking because they found a virus which was very, very genetically distinct, very different in nucleotide sequence from the viruses that had been previously isolated. So the previous viruses that had been isolated were kind of like runners on 125th Street, but what they found was actually kind of a runner out, in Brooklyn with this new sequence. So what's really exciting is that because they found this runner off really far away, they actually realized that the race must have been going on for longer, basically. So they figured out through this new sequence, having data on this new viral sequence, that HIV has been in the population not since the 1940s or 1950s, as was first estimated, but much earlier, about 1900. So that's one way that viral data can actually be used to estimate when some of these events actually occurred.
Would you describe other examples of how this approach has been used?
Another example is a story or an event that happened a couple of years ago. There were these 5 Bulgarian nurses in Libya, and they were accused of infecting children at this hospital with HIV and with hepatitis C virus. And the Libyan government was putting them on trial and would possibly give them a death sentence. So some evolutionary biologists contacted the children that were infected with HIV and hepatitis C virus. Some of these children went over to Europe afterwards to be treated, and so they contacted them and sampled the virus from them And from those very diverse samples, they determined the time at which these individuals were most likely infected. And so from that diversity of samples, they figured out that they must have been infected before the nurses even showed up at that hospital. So that's another way to use viral sequence data to understand or get an idea of what happened historically without having direct data from that time. A third example, besides the HIV example with Libyan nurses and the more recent story with Michael Warabee's HIV work, is a story about these cougars in the western United States. So these cougars in the western United States are infected with something called FIV, feline immunodeficiency virus. It's related to HIV, only it doesn't cause any symptoms for the cats. So researchers put out a question. They asked, well, what has been happening to the cougar population over the last few decades? Have they had population lows? Are their populations increasing now? So what can we actually understand about cougar population sizes in the past? So what they did, they didn't have any direct data on it, so they didn't know. So what they decided to do was they decided to actually look at, again, viral sequences, current viral sequences that these cougars had. So they went out and they sampled these big cats, and they sequenced their viruses. And because these viruses mutate very rapidly, they can be used as this clock to date certain events. And what they found out was that in certain regions, the viral population was genetically not that diverse, and in other populations, it was also not that diverse, but it was different. So what they were able to show was through looking at these sequences that there was really this geographic separation, and that the cougar population went through a significant population low. And that's why these genetic sequences of this virus were very different in different areas. So those are three examples, very clear examples of how viruses can be used for us to really get at an understanding of what has happened historically without actually having direct data from there, because we're really inferring it from these viral data.
Will you please tell us more about your research?
One thing that I find very interesting with respect to these viruses evolving so rapidly is that for some viruses, for example, also with measles, a lot of these changes don't have any kind of they don't change the face of the virus. So those can be used for certain things like reconstructing historical events, trying to find out what happened when, etc. There's another class of mutations where the mutations to the virus actually do alter the face of the virus, and it's really the face of the virus that our immune system sees. And so for things like influenza, the virus is very rapidly evolving as it goes through the human population, and sometimes those changes allow for escape from immunity. And so a mutation results in a virus which then has a lot of individuals who are susceptible around it, and so that virus can actually spread much more quickly than a virus that had a lot of individuals in the population that were already immune to it. And so trying to understand through incorporating genetic data, trying to understand how viruses actually can escape from immunity is I think very interesting. And then how viral evolution actually affects how quickly a disease spreads and how many people get infected over time. So that's another aspect of viral evolution, which is very interesting and which I'm focusing on in my research.
What do you enjoy about your work?
So what I enjoy, I think, about my research most are a number of different things. One is that you can use a lot of different tools from different subjects to address problems in public health, and specifically with respect to viruses in the population. So I use a combination of mathematical approaches, and I use statistics, and also concepts from ecology and, of course, from evolution. So all those things, all those subjects have to be integrated in order to be able to understand things like how quickly a virus passes through a population, how quickly it evolves, and things along that line, how much of a disease burden it produces in the population. So we need to actually have a very interdisciplinary approach to understanding these patterns. The second thing I really enjoy about the research is that diseases and viral diseases are a really great model system to try to address questions that are open in the field of ecology and evolution. Specifically, there's a lot of research going on right now in what is called contemporary evolution, which shows that that evolution occurs very, very rapidly for many organisms. So that includes things like the finches that Darwin studied. They evolve very rapidly, even on the order of decades. And a lot of other organisms, such as salmon, are known to evolve very rapidly, especially when you think of fishing pressures and selection coming from those fishing pressures. So there are all these different examples of how evolution in organisms is occurring very, very rapidly on the order and timescale of decades. So I think viral diseases, provide another really great example of this, where viruses that are evolving over the timescale of decades oftentimes allow for individuals to get reinfected because they're evolving with respect to their being able to escape immunity of the host population. So there's a really nice interface, I think, between a lot of ecological and evolutionary topics and topics which are really at the interface of public health.
For more information about this story, including links to primary and popular literature and classroom resources, visit the National Evolutionary Synthesis Center website or the Understanding Evolution website. More stories are available in the Evolution in the News archives on either site. The National Evolutionary Synthesis Center is funded by the National Science Foundation to promote research in biological evolution.